Hello everyone, and welcome to today's webinar, Fast Track Your Biologics and NGS Workflows with Automated UVVis Quant on Lunatic. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Unchained Labs. To learn more, visit unchainedlabs.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers, Nelis Dennis, Product Manager, Lunatic and Stunner at Unchained Labs, Phylicia Dassardo-Joseph, Scientific Investigator, Biopharm Discovery, GSK, and Matt Humes, Senior Lab Manager, Lab Automation, Beam Therapeutics. Nelis, you may now begin your presentation.
Hi, thanks everyone for joining us in this very exciting presentation. Together with our two external speakers, we'll show how you can fast-track your biologics and NGS workflows with automated UVVis Quant on Lunatic. I want to start with the most famous and most successful example of automation ever, laundry. When doing hours of work with way too much soapy water, everyone was ready for a better way. For sure, when the piles of clothes just keep getting bigger and bigger. For quantifying nucleic acids or protein, I'm sure everyone remembers the spectrophotometers, where you could only load 1 centimeter cuvettes that required at least 100 microliter sample and read one at a time. But like with the laundry, UVVis spectrophotometry has evolved to handle a big pile of samples, all automated and using less volume and time. Our Lunatic is a full-spectrum UVVis spectrometer, which requires only 2 microliter per sample and no dilution steps nor standard curves. Lunatic can measure up to 96 samples at a time in just 10 minutes using our plates with 2 microcuvettes, which allow reading higher concentration samples. If you have samples with a low concentration, we have a second plate with 1 microcuvette, and the 96 samples will be read in just 5 minutes. Our plates have a standard SBS footprint, so they're compatible with liquid handling robots. Now, how does our Lunatic do this? Well, Lunatic is the only system out there that can measure nucleic acids and protein at high throughput and high concentration. The microfluidic circuit with 2 fixed path lengths, as you can see highlighted here, covers a wide dynamic range of 0.03 to 275 OD. The unique microfluidic circuits molded into Lunatic plates ensure that there's no cross-contamination or evaporation. Fixed cuvettes make for accurate and fast concentrations, since we can always be sure of the path length used in Bayes' law's calculations. It also allows us to correct for stray light and other background. Fixed path lengths mean you know exactly what you're measuring. Now let's take a more in-depth look at how it works. Samples are deposited into the input well and drawn into the storage channel by capillary action. A sample can be left in the storage channel for up to 2 hours without detectable evaporation. After the plate is loaded into the instrument, the empty microcuvettes are read first, and that will remove any absorbance contribution from the plastic. And then a pump applies a small vacuum which moves the sample into the microcuvettes for reading. Now, high Lunatic plates have 2 microcuvettes in each circuit with a 0.1 and a 0.7 millimeter pad length. The 2 different sized microcuvettes make the wide dynamic range possible. Lunatic has an accuracy within 2% and a precision within 1% CV. This can be verified with the fundamentals, or third-party certified standards. 5 tryptophan standards at different concentrations to check accuracy, precision, and linearity. Now here's a look at linearity, accuracy, and precision. This shows an IgG dilution series of 8 replicates from 2 micrograms per microliter to 200 micrograms per microliter. The close agreement between the target concentration and the average measured concentration shows off Lunatic's spot-on accuracy within 2% of the expected value, with an R-squared value of 0.9999. The low CV values across the dilution range help show off its precision. And the data shown here demonstrates how Lunatic generates the same amazing results for genomic samples. The highest concentration of DNA in this dilution series was 12.8 micrograms per microliter. The R-squared value for the series showcases the high linearity across the huge dynamic range of Lunatic. So just run your samples at the concentration you're already working with. Don't add in extra dilution steps you don't need. Here you can see a plot with 3 concentrations of DNA. Either intra-instrument, ran in octuplicate on the same system, or inter-instrument, ran in octuplicate on 8 different Lunatic systems, for a total of 64 samples per concentration. On one instrument, we've already discussed the amazing results earlier in this presentation. Now here we can see that with 8 completely different systems, there's also nothing to worry about. Having systems in different labs, in the same building, or even at different sites globally will always provide the same high-quality results. And the features don't stop there. We have IQOQ procedures available, and for labs doing manufacturing, quality assurance, and quality control, Lunatic now has a performance verification for new instruments. These tests with certified cuvettes meet the United States Pharmacopoeia and the European Pharmacopoeia guidelines for UVVis spectrometers. The tests and all necessary calculations are completely handled automatically by the software. And now, I'm very glad to introduce you to our first external speaker. Matt Humes is the Senior Manager Lab Automation at Beam Therapeutics, based in Cambridge, Massachusetts. He gained his bachelor's in biochemistry at the Stony Brook University of New York and is working on his master's in bioengineering at Harvard. Previously, Matt built his expertise as supervisor automation at Regeneron Pharmaceuticals, where he installed more than 30 liquid handlers, 5 fully integrated systems, and 3 biobanks. Within his current role, he oversees Beam Therapeutics' lab automation and robotic efforts. Without further ado, I'll gladly hand it over to Matt now.
Thank you, Nelis, for that introduction. I am Matt Humes, and it is my pleasure to share a little bit about Beam today and some of the automation we've been able to set up around our primary workflow. Beam is a relatively new biotech company. We came out of stealth mode about 3 years ago and went public just about a year ago. What we are is a next-generation gene editing company. At this point, everyone is pretty familiar with CRISPR. When talking about CRISPR, the common analogy that gets used is molecular scissors, where they find a specific site, cut the double helix, which paves the way for cool things like deletions or insertions of new DNA. But sometimes you don't want to cause that double-stranded break, and that's where the base editors come in. If we continue the molecular scissors analogy, we are like a pencil and eraser. We can go in and specifically change single base pairs within the genome at a precise location without inducing a double-stranded break. So here is a very high-level view of how we do that. Essentially, we use a dead or modified Cas9 molecule and the same guide RNA that you would use to target a particular site, and we tether a chemical modifier to that complex. That modifier can come in and change a specific base by the chemical modification that you see on the right side of the screen, deaminations that convert C to T or A to G. This is very high level, of course. If you're interested in learning more about the specifics, please check out beamtx.com and you can find links to the papers and data behind this awesome technology. As you can imagine, being a base editing company, there's a large amount of next-gen sequencing. Here is a very high-level view of one of our in vivo workflows. Our base editor is paired with a delivery mechanism, in this case a lipid nanoparticle. Animals are then dosed and livers are harvested for extraction. Here's where it goes onto our automated system, which handles all of the purifications, normalizations, PCR and cleanup steps, pretty much everything up until it is loaded onto the MySeqs. So here's the journey of how we got there. When I was hired back in 2018, pretty much everything was manual. You can see from the cartoon, we couldn't afford one of those cool PCR robots, so we got an undergrad and a cardboard box. Those of you with automated systems can probably remember these times. Many of us were probably that undergrad in a box at some point, which is pretty much how all automation projects start. We purchased a liquid handler to address the tedious and difficult sample transfer steps, but quickly realized that this is just one small piece of the entire process. We found ourselves coming back to the system over and over again to perform simple tasks like sealing a plate, centrifugation, or bringing a plate over to a reader. While a step in the right direction, it was not particularly groundbreaking. High throughput days pretty much entailed just bringing the reader closer to the liquid handler. So we decided to move to a fully integrated system to address these moments with the goal of creating large blocks of walkaway time. Here are the broad metrics we consider necessary for a device to fit into our system and workflow. First, it has to be high throughput, which means not only fast, but formatted in a way that it can keep up with our purification process, which is done in 96 well plates and streamlined enough to handle 10 to 15 plates per day with a high rate of success. Next, the device itself needs to be operated autonomously by a scheduling software. We need to be able to load, read, and unload the right plate at the right time and send that data forward through the process. Also, run parameterization was key for us. We never do the same thing twice. The conditions we read at, the number of samples, how we blank, can all vary from run to run, even within a single run from plate to plate, and we need to be able to pass that data to the instrument programmatically, so that when we execute the read, we know it is doing exactly what we intend. Lastly, once our software has loaded the right plate and the right parameters were sent, we need to be able to trust the data. Finding an instrument that we can rely on to perform the same way each run was vital to our process. We looked at many of the existing devices and chemistries out there through the lens of our workflow and the instrumentation that we had on our particular system. While all of these methods work, they each had drawbacks. Fluorescent assays require additional sample prep, they are dependent on the performance of standard curves, and they utilize light-sensitive reagents. It also gives no qualitative data on the sample itself. Off-system devices created a huge gap in our data continuity. In addition to being slow, we did not want USB sticks and file drops to be our main source of data transfers. Finally, there are what I call semi-automated solutions, which internally have automation-friendly specs, such as well spacing, but the tray itself tends not to be sealable or movable by most robotics. For us, the Unchained Lunatic was the answer. It has a feature-rich API that allows us to take the data we get from LIMS or sample scans on the system and create experiment parameters on the fly, as well as accurately labeling data that comes out of the device so there is no gap in traceability. Another pro for this is that there is no sample prep. There are no time-consuming serial dilutions or light-sensitive reagents to program around. All you need to do is load a few microliters onto their plate. Not only does this lower the cost per sample, it frees up robot time and reduces those precious robot consumables that are very hard to come by these days. The Lunatic plate itself adheres to automation standards, making it easily handleable by robotic arms and liquid handlers. And lastly, it is quite fast, with a full plate read around 10 minutes, partial plates significantly less than that, and it is able to be used in a GLP setting. Here is a high-level view of a typical 10-plate and 8-hour day. On the Gantt chart, the dark green blocks represent DNA purification steps, tan blocks are points where we need to quant samples, and blue is our NGS library prep consisting mostly of PCR and cleanup steps. So without integration, what does this look like for our undergrad? As you can see, within an hour, our technician has to come back to the system 4 separate times to take quants offline. Throughout an 8-hour day, this number goes up to 20. What's really staggering is this is just one assay. Scale this up to multiple assays on multiple systems across an entire week, and you can imagine the time that is lost. By choosing a device that has met all of our integration criteria, we are able to remove all of these touchpoints. Instead of returning to the system 20 times throughout the day, our team can walk away for the entire length of this 8-hour window. freeing them up to spend more time designing and executing experiments and really pushing the science here at Beam. And perhaps most importantly, more time is freed up for coffee. As I mentioned before, this is a typical 8-hour day. The Lunatic allows us to make this an end-to-end solution, which allows us to kick off another set of these 10 to 15 plates before our team goes home for the night, extending the workday even further. And now for my favorite part, here's a short clip showing all of this in action. From automated storage to sample loading and reading, all made possible by the integration of the Unchained Lunatic. Alright, so thank you everyone for coming to hear Beam's automation story, and thank you to our partners at Unchained for letting me talk with you all today.
Thank you, Matt, for that great presentation. I fully agree, more coffee breaks is always better. Now, our next speaker, Phylicia, will talk about her protein workflow with two of our systems, Lunatic and Stunner. Now, just before we continue, I would like to give some background info on Stunner. In essence, Stunner is our UVVis technology from Lunatic in combination with dynamic light scattering. Stunner is the first and only instrument that has both full spectrum UVVis and dynamic light scattering all in one. The combination of these two detection methods provides a more in-depth analysis of biological samples. Stunner provides information on the concentration, size, and polydispersity. Stunner does all of this with its unique Stunner plate that only needs 2 microliter per sample. So low volume, high throughput DLS to get the size and PDI data you need while quantifying protein concentration. All this is done in a 96-well plate-based format that powers high throughput analysis, giving you all this data on 96 samples in an hour. And now, I'm very glad to introduce our second external speaker. Phylicia Dassardo-Joseph is a scientific investigator within the Biopharm Discovery Department at GSK. She gained her bachelor's in biological sciences at the University of Leicester. She has 7 years of experience working at GSK and built her expertise in biophysical characterization, biopharmaceutical developability, crystallography, high-throughput screening, and automation. She has successfully developed and embedded automation of several manual processes via miniaturization, use of liquid handling robots, and scientific knowledge. Within her current role, she is advancing automation and data capabilities to further support streamlining of early biopharmaceutical drug discovery processes. Phylicia, the stage is yours.
Thank you, Nelis, for the introduction and for the opportunity to talk about our UVVis protein quantification workflow using a liquid handler system which is integrated with a Lunatic. The agenda for my presentation today is as follows. I'm going to give a high-level overview of our workflow, the past and present state of why we decided to proceed with an integrated system, how the liquid handler and Lunatic integrated system performed UVVis protein quantification, and finally, an example of our assay characterization where we use the Stunner for DLS. For the overview of our workflow, first we receive a panel of purified proteins which requires screening. The liquid handler integrated system is used to perform all sample preparation. A part of this process is to perform a UVVis measurement via the Lunatic to quantify the concentration of our proteins. Using the outputs from this measurement, the system will calculate the new amount of sample and buffer required to normalize the sample and generate a new concentration. New concentrations are required throughout our screen in order to analyze our proteins by different characterization assays. So where did we begin? Previously, we would need to perform all sample preparation manually using two different systems for UVVis quantification, depending on the concentration we were trying to achieve. One was used for concentrations up to 20 mg per ml, which was faster, and the other was up to 150 mg per ml. Both systems were manual, and it was possible to measure one sample at a time. If there was a larger panel of proteins, this was challenging and often required repeats due to failed readings or high percentage CV observed between replicates. We wanted to increase the speed at which we could perform our UVVis measurement that was a high throughput and low sample requirement. This system would ideally be automation friendly, such that the system would be future-proof. The Lunatic was a product that looked like it could fit this gap in our process, so we decided to investigate it and compare it to our current systems. During our comparison, we identified that the Lunatic was comparable to both of our systems up to their respective concentration range. We decided to purchase the system and investigate the possibility to integrate on the liquid handler system. What we managed to achieve was a system which was fully automated that could perform our UVVis protein concentration workflow. In the video, you will see the liquid handler mixes the samples before aspirating and dispensing into the Lunatic plates. Once this is complete, the Lunatic door will open and the gripper arm will transport the plates into the Lunatic. Once the door is shut, the measurement will begin, and this will take some time depending on the protocol and the number of samples that we're running. For today's purposes, I've skipped the wait time. Once the measurement is complete, the door will open and the gripper arm will retrieve the plate. It will then shut the door and the workflow will continue using the results from the Lunatic to generate new samples with a different concentration required for our assay characterization. For instance, DLS. For our assay characterization, we perform dynamic light scattering to measure the size of our protein in a solution. Previously, we were performing this manually, and the DLS assay required large amounts of sample. Therefore, we investigated methods to miniaturize this assay to reduce our overall sample requirement. We already knew the stunner plates were amenable to automation as we used the Lunatic plates in our protein concentration workflow. We compared the stunner to our legacy DLS system, which showed reproducible results, where the legacy system is shown in orange and the stunner in the blue. The shapes are pairs of the same sample. This showed we could produce the same data quality as our legacy data using 90% less sample, and now we are routinely using the stunner for our DLS workflow. To summarize, we have integrated the Lunatic with our liquid handler. This has allowed us to increase our sample preparation efficiency and speed, reduce our personal resource since we can walk away during sample preparation, and enhance the throughput of our screen. For the stunner, we have reduced our sample requirement by greater than 90%. Thank you for your attention. I hope you enjoyed this presentation, and I'll now hand back over to Nelis.
Thank you all for attending our presentation. Matt, Phylicia, and myself will take any questions now.
Thank you, Nelis, Matt, and Phylicia for your informative presentation. We will now begin the Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, so Nelis, this question will be for you. Can this method be used with RNAs?
Yes, that's the short answer. Our system has applications for DNA, RNA, any kinds of protein. So this also works with all the integration steps. So yes.
Thank you, Nelis. And I've got another question coming your way. What's the concentration range of Lunatic?
So the Lunatic has a concentration range of 0.03 OD to 275 OD. That translates for DNA double-stranded, it would translate into 1.5 nanograms per microliter up until 13.7 milligrams per microliter. And for protein, about 0.03 mg per mil to 275 mg per mil.
Thank you, Nelis. Matt, this question is for you. How many samples can you handle in a day? 10 plates of 96 as shown on the slides? Was this possible with your former method?
Sure, so that's a great question. The previous method, we were closer to 2 to 5 plates, and that was a very busy hands-on day. With the integration, our average tends to be about 10 plates, which allows us to get through the entire NGS workflow in the same day. On heavy days, we've actually been able to do 15 plus plates without any slowdown. The bottleneck tends to be more of the extraction process. The Lunatic is quick enough to keep up with as much as we throw at it.
All right, thank you, Matt. Phylicia, this is for you. How much is your screening throughput enhanced with Lunatic? And you can give me just a ballpark number.
So I would say our screening throughput has enhanced by at least 5-fold by Lunatic integration.
All right, that's amazing. Thank you. And it looks like we have one more question for Matt and Phylicia. How difficult was it to actually integrate the Lunatic?
Sure, I can start. It was actually very simple. The device itself was designed to not only look good, but actually be very integration friendly. The flat face of the device allows for a lot of robot access, so you have a lot of options as to where you put it. The other thing, like I mentioned in the slides, was the plate itself is SBS format, so it allows us to use any kind of robotic arm storage or liquid handlers. For the most part, if you can handle a 3 to 4 well plate, I've found that you can handle the Lunatic plates just fine.
I would agree with Matt as well that the integration was pretty easy. I think overall it probably took us about a week plus to integrate the system and then plus start having our protocols and workflows in place. We had a similar system before that we had integrated, so the predecessor didn't take the drops, and so I think that made it a lot easier as well. And then in terms of communication, everything works over a network rather than an Ethernet cable. So I guess that makes it nicer in terms of being wireless.
All right, thank you again, Matt and Phylicia. And thank you to all of our speakers, Nelis Dennis, Matt Humes, and Phylicia Dassardo-Joseph for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Unchained Labs, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.